Okay. Hi, I'm Kailash. I'm final year dual degree student.
Just Kailash, before you start, why I'm seeing not seeing the full screen? I'm seeing Swagatika's iPhone, Karthik Raman, anybody who's talking coming on the sidebar. Why is that?
I think that's gone now. No, I think that's gone now.
No, it's still with me. I'm like seeing a very tiny screen now. Even though I have full screen, my screen for the presentation is very tiny.
Can you still see it?
That's who the audio is coming from.
You could see the full screen, Imanshu.
Really,
Apart from the full screen, there's a smaller screen.
what
That's
I've done.
who's the source of the audio that you're hearing.
Should I change something?
No, just go ahead. Just go ahead. It's weird. Yeah.
I don't know what I've done.
For me, it's the full screen.
Himanshu, you need to scroll through.
I was just sitting without touching anything, and it just happened. So I don't know.
Okay.
Continue. Let's start. Let's not waste time on that.
Okay, I'm Kailash. I'm a final year dual degree student working with Professor Manikandan Narayan on this project. And the title of this talk is tissue level differential correlation analysis of Alzheimer's disease. And can somebody confirm if they can see the pointer?
Yeah.
Okay, so this is the a representation of a normal healthy human brain, and this is how the human brain changes over time when one person is affected with Alzheimer's disease. So as you can see here, there's a lot of neurodegeneration happening, and this neurodegeneration is what leads to shrinkage of the brain tissue here. So this is brief intro to what happens in the area. I'm trying to see. Okay. So a brief intro on Alzheimer's disease. So Alzheimer's disease is neurodegenerative disease, and it is marked by the two major misfolded proteins, which accumulate either inside the neuron or in the vicinity of the neuron, which causes the neurodegeneration pattern that we saw in the earlier slide. So as you can see here, there are some stages which have been characterized by previous studies where they characterize the spread and how severe the disease is. And the main point to take away from this slide is that this is not Alzheimer's disease is not restricted to one particular tissue. It travels through, and the degeneration happens starts at one particular region, and then it spreads throughout the brain. And so this points to the need for analysis of multi-tissue gene expression data to even understand what are the genes that are involved and how this disease progresses. So the main questions of interest that we are focused on is, are genes in different brain tissues functionally coupled or correlated across healthy individuals? And can we see? I'm hearing lots of noises. Okay, so I'm trying to understand the differences in gene expression across healthy and disease samples, and whether they are functionally coupled or correlated, and how does this correlation gain or loss in the disease. And Alzheimer's disease, rewire this network of intertissue correlation with the progression of. So as far as I understand, most of the AD gene expression studies are focused on single tissue analysis, and to actually provide further understanding into such a degenerative disease specifically, it's necessary to understand the intertissue distribution from multiple regions of the brain. So this is the lateral view of the brain regions under study. And here we see that in the study we are studying 4 different brain tissues or brain regions, namely the parahippocampal gyrus, inferior frontal gyrus, superior temporal gyrus, and frontal fold. It is ordered based on regional vulnerability to AD. So previous study which is published by Mount Sinai, this particular paper here, it has provided us with this data, and they have also come up with a method to understand which region of the brain is most vulnerable to AD, and that is how this has been arranged. And so there's a mapping of the brain regions to some labels called broadband areas, and we will be looking at these 2 brain regions, namely BM36 and BM44, in this study, in this particular talk. So BM36 is the most vulnerable to AD, and BM44 is the next most vulnerable to AD, based on 19 brain regions that the Mount Sinai cohort studied. So yeah, this slide will so this explains what I'm trying to do. So what I'm interested in to look at is inter-tissue progression of disease and how it varies between control and AD. So I'm focusing on one particular gene that says G1, G2, and G1 is from tissue A and G2 is from tissue B. And I'm looking at the same gene pair in the disease A. And similarly, it's the same G1 and G2, and G1 again here is from tissue A and G2 is from tissue B. Now, initially what I do is I find correlations between such gene pairs, all possible gene pairs actually, between these two brain regions, and put a FDR cutoff and take genes which are correlated. And similarly, I take correlated genes in AD. And now I have a correlation value between G1, G2 in the control case and G1, G2 in the disease case. And then I find a differential correlation. Basically, if you could talk of it to make things easier, it could just be the difference. For example, if this is 0.9 and in the AD case it's 0.7, so the difference in correlation is 0.2. And such significant differences in correlation are considered, and gene pairs with such significant differences in correlation will help us would contain the most of the information in understanding the disease. So we take such gene pairs with, which are differently correlated, and now we are trying to understand why is there a difference in correlation. So understanding will point towards the differences in the gene co-expression, which leads to it becoming AD from control, and assigning factors to it will help us in understanding the causes or the different methodologies that work behind this differential, this gene expression pattern which is observed. So first, preliminarily, what we are doing is we are just differentiating it into two cases, where case one, which is expressed stage, is gene differential correlation, which is expressed just because of cell type frequency, and Y percentage is everything else. So what do I mean by differential correlation, which is explained by cell type frequency? So let's say there is one gene 1. This gene 1 marks a particular cell type, say CT cell type 1, and with increase in cell type composition of this cell type, there will be increase in gene expression of G1. So similarly, if G2 is a marker for cell type 2, higher the frequency of cell type 2 in the particular tissue, higher the gene expression. So just because the cell type frequencies vary between these two cases, we say between control and disease here, there could be a differential correlation with this observed. And we'd like to identify these changes separately apart from changes which are due to other factors, which could be internal regulation of the gene pairs, which leads to disease progression. So this the data for this study is obtained from Mount Sinai cohort study, and we are the AD samples are the definite AD, and there are 68 subsamples for BM36 and BM44 regions together, and for control there are 53 samples for those two brain regions together. So this number varies from this number because there were 4 tissues which are considered, and when I consider a pairwise disease correlation between two tissues, I take only the samples whose gene expression exists for both of the tissues. So, I did so when we do this analysis, so after following the methodology in the previous slide, we identify 18,942 significant DC relations which span across 8,264 unique genes. So number of genes initially in the study are 21K odd. So 8,264 of those genes are involved in relationships with differentially correlated at the 1% FDR. So one particular example is here between ARCC1B and LDLR-A-depot. So here we see that initially in the control case, the gene expression between ARCC1B and LDLR-A-depot is negatively correlated, and with the disease case, it becomes positively correlated. So there is this is what I mean by difference in correlation. So something that was negatively correlated becomes positively correlated, and the extent to which the gene expression pattern changes means that that particular gene pair, gene edge is differentially correlated could point to. So it's more important to look at such gene pairs. So similarly, like I said before, 18,942 such relations identified. And so now we are trying to understand why there is a difference in correlation. So that would be the current analysis that I'm doing. So why is there a difference in correlation? It could be, like I said before, just because of differences in cell type frequency, and just because of that, there could be differential expression noticed, and we are trying to figure that out right now. So main conclusions are that multi-tissue study of such neurodegenerative diseases is essentially important and to understand the disease processes involved in such neurodegenerative diseases. And preliminary analysis, which I have not discussed here, points towards the fact that most the differential correlation in AD which has been observed cannot simply be due to the changes in cellular composition. So, like I said before, the X percentage which I talked about, where the X percentage accounts for the percentage of the differential correlation, which can be explained by cell type frequency alone, does not explain all the disease, all the differential correlation. So there are other factors in play, and now we are trying to confirm these preliminary findings in published single cell data to further strengthen the hypothesis, and trying to look at further studies to interpret the DC relations where there is intra-cell regulation, regulation of DC, not just because of cell type frequency, but because of other factors. And other factors is also very ambiguous. So we are trying to make it more less ambiguous and trying to figure out more like cell type frequency is one particular thing. So we are trying to delineate other factors into multiple sub-factors, and hopefully this will lead to better understanding of it. So yeah, thanks. So I'd like to acknowledge Sai Kumar, Srivatsan, PG, and Mani sir for we all work together in this project, and BERS group, which is the lab group of Professor Manigandan Narayan, IBSE-NCCBR. Yeah, thanks. Are there any questions? Am I online?
Yeah, I think everyone's just, tired. Long day with no tea, as Himanshu was mentioning.
Yeah.
Could have arranged for tea and vada, Karthik.
Yeah, we'll do that once, Swiggy and all becomes functional.
Virtual, like Ravi does.
Oh, virtual. I can arrange anything then.
Yeah, exactly.
Some exotic ice cream, if you will. It's getting warm.
You can
Okay.
send the catalog and say choose
So
and rule.
one thing that may be of interest here to a larger audience, I mean, even if we are interested in other topics like beyond Alzheimer's, is we found this linear regression model very recently in a paper called Cell Code, where they try to decompose the expression of a bulk tissue into the expression at a cell level, like single cell level at a cell type level, and then multiplied by the frequency of these different cell types that composes this tissue. And then it decomposes into these other factors. So in this particular case, there is this X and Y percentage. X is due to cell type and Y is due to other factors. And it basically, that model gives you the changes in some disease-induced changes, either at the expression level or at the correlation level that is due to cell type frequency, that will be your X. And then due to the internal cell changes, that will be Y. And then the other factors, which will be Z. And so whenever you have some kind of a bulk data that's coming from a tissue and you want to understand it and you want to decompose it into the different cells. I know this is also very popular in cancer when you're looking at the microenvironment of cancer, and you get your measurements from a lot of immune cells and the cancer cells from the cancer, and then people try to deconvolve this mixed signal into the individual signals. So some of these methods could be of use. So I just wanted to point that out so that you could think about it here. Thanks.